Thank you very much for a great introduction. I'm Seiya Imoto. I'm very happy to be here, and also thank the staff of this symposium. And five talks are related to our human genome. However, I am focusing on the genome analysis of our commensal bacteria. And this is become a very famous neighbor of us, the intestinal microbiome or microflora. And in our intestine, there are various kinds of bacteria and many number of bacteria living there. And this is also called microflora. So flora means flower garden. So various kinds of bacteria live in the intestine. And they play a very important role to maintain our health condition. For example, they promote a breakdown of the sugar and the synthesis of the essential vitamins or amino acid to keep our life, and also improvement of the intestinal environment by producing the short-chain fatty acid with immune modifying effect. And I would like to explain what metagenome analysis is. So we are here. So we are here, and we are interested in our in the bacteria living in our gut. And usually, so the first bacteria was discovered more than 300 years ago. So long history the bacterial research has. However, the traditional research style of the bacteria is to isolate our interested bacteria and culture. Then the various wet experiment performed to isolate cultured bacteria of interest. However, the way of researches was changed dramatically by using the next generation sequencers. So we can extract the DNAs in the sample, and all DNAs can be sequenced by the next generation sequencer. So we can know that what sequences exist in the sample by the data, short-read data. So the length of the DNA sequence is just around 200 base pairs or 300 base pairs, very short. However, we can identify these DNAs comes from, for example, E. coli or other bacteria. So the game is changed. The traditional method for bacterial research is just rote fishing. However, using the next generation sequencers is similar to roll net fishing. So all DNAs can be analyzed. So you can imagine that the bacteria is hard to culture, how to isolate it, how to culture cannot be investigated by the traditional method. So we can get a fragment of the bacterial DNAs. However, we can classify, we can put the annotation to each of DNA fragment using the computer and the databases. So we can find the composition of the bacteria in our gut or stool sample. So you can find that his intestinal bacteria, so around 15% of them are Bacteroidetes, or 10% of bacteria are categorized into a Faecalibacterium, like this. So using the next generation sequencers, we can find the whole picture of our commensal bacteria. And this slide compares the information encoded in our human genome and the bacterial genome. So we are made by 40 trillion of cells. And in each cell, 20,000 genes are coded. However, in our intestinal bacteria, the number of cells is 100 trillions. And on the bacterial genome, 10 to 20 million genes are coded. This is more than 100 times larger than the number of human genome. So enzymes that cannot be produced by human genome, but can produce bacterial genome, those play a very important role to keep our life. So the number of cells and the genes of intestinal microflora are much more than those of human genome. So our intestinal microflora influences on our health, diseases, mental status, and so on. And nowadays, it is very trivial that it is not enough to know that our health condition or disease by knowing our human genome, but necessary to know our commensal bacteria. Microflora is called alter ego. But not all bacteria are beneficial to our health. Someone play a bad effect to our health. For example, E. coli, some kind of E. coli produce a toxic element, and it leads to a very severe pain. But even for healthy people living in healthy, but they can have pathogenic bacteria in their intestine. This is called a pathobiont, and Professor Shibata introduced Helicobacter pylori for gastric cancer. This is one of the examples of the pathobiont, the healthy people but has Helicobacter pylori. It leads to gastric cancer in a high probability. But the research is about the pathobiont is now just started, and we need to investigate the long effect of the pathobiont in some cohort. However, if the pathobiont is harmful to our health, we want to eliminate, like Helicobacter pylori. But if we eliminate some pathogenic bacteria in our intestine, we usually take antibiotics. However, the antibiotics cannot target only to pathogenic bacteria, but can kill beneficial bacteria or optimistic bacteria. So this is the example of the strong antibiotics taken prescribed to mouse. So the composition of the intestinal microflora dramatically changed. This is a 0 week, just after taking the antibiotics. There are many kinds of bacteria there. However, after the 2 weeks, the only proteobacteria, this include E. coli or Klebsiella. So some dangerous bacteria are included in this group. They can survive from the antibiotics. But after stopping the antibiotics, the diversity of the composition becomes larger. Larger. So this data shows that antibiotics can kill many kinds of bacteria, not target to pathogenic bacteria. Pseudomembranous colitis is a typical disease occurring by taking strong antibiotics. So this disease, we know the cause of this disease. He is Clostridioides difficile, this bacteria. Even healthy people, more than half or 80% of people has Clostridioides difficile in their intestine, but a very small portion. However, he is drug-resistant bacteria, and a patient who take a very strong antibiotics. So his commensal bacteria, many of his commensal bacteria died. However, the Clostridioides difficile can survive under this condition. And then Clostridioides difficile produce toxic element. It leads to a very severe inflammation like this. So in this case, we want to eliminate Clostridioides difficile from the patient intestine. However, it is very difficult because Clostridioides difficile is a drug-resistant bacteria, so antibiotics doesn't work. So this is US CDC report in 2019 before Corona. So this number of deaths occurred in US. So the right part is a Clostridioides difficile cases, and the left part the cases of antibiotics-resistant bacteria. So the Clostridioides difficile is a special. The cases by a Clostridioides difficile are counted separately, other than the other antibiotics-resistant bacteria. So the recurrent Clostridioides difficile infected patient, there are no efficient antibiotics to this disease. So FMT, fecal microbiome transplantation, the FMT in short, is performed to such patient. This is surprising treatment. The microflora is taken from the fecal, from the stool sample obtained from the healthy people, and the transplantation was will be performed to a patient. But there are we don't know the mechanism of the efficacy of the FMT. However, it is reported that the very high efficacy, the 92% patient, their condition of the intestine improved, but the number of patients not so large in this paper, clinical research, and also we don't know the healthy microflora. There are no definition. FMT is performed under the just naive idea that the healthy people's microflora should be good, but no definition. But unfortunately, in 2019, 2 deaths were occurred by infection of the pathogenic bacteria comes from the donor sample. So donor has pathogenic bacteria in their microflora, but they live healthy. This is a mystery. However, if we sequence a microflora from a healthy people, from 10% to 20% people, we can find pathogenic bacteria's piece of genome in the metagenome data. So this is not a surprising news for us. But there are no alternative therapy for difficile infection patient other than the FMT. So FDA restarted FMT maybe in the 2020. But some very impressive papers were published in high-impact journal like Science, and they reported that the efficacy of the immune checkpoint inhibitor can be strengthened by combining the FMT for, I forgot, melanoma, yeah, for a melanoma patient. So we want to know the mechanism of FMT, yeah, how improve the condition of the intestine by FMT. Yeah, if we know the mechanism of the FMT, so we may create alternative way by mimic the FMT. So we have the metagenomic data of the patient before FMT and after FMT, and the metagenome data of the donors. So we have the 3 data for one patient. So you can see that before FMT, the patient microbiome, the gamma proteobacteria are dominant. It includes E. coli and the Klebsiella. But after FMT, so you can see the center bar plot. It is very similar to donor's pattern. So we received the stool sample of the 9 patients from the Harvard University, the Brigham and Women's Hospital, and we performed the sequencing analysis and the data analysis, and obtained this result. So 9 patients before FMT and after FMT and donors composition of the bacteria. So we can see that the changes of the bacterial composition. And after FMT, the pattern of the bacteria similar to the donor. And before FMT, the dominant is gamma proteobacteria. But from this data, we cannot reach the point of the drug discovery. So mechanism, we cannot find the mechanism of the FMT by just looking at the composition of the bacteria. So we further investigate not only bacterial species, but also the genes on the bacterial DNAs. So from genes, proteins are produced. And the bacterial proteins play a very essential role in our pathways. So the pathway is consists of the multiple chemical compounds and the enzymes that realize a higher level of function. For example, amino acid production or vitamin synthesis was like this. So the pathway analysis was performed, and we compare the data of the before FMT and after FMT, and representative pathways were discovered. For example, fluorobenzoate degradation was activated only before FMT, but inactivated after FMT. And these red three pathways were activated after FMT. So those three pathways, the two pathways can be interpreted by existing literatures. However, the non-homologous end joining pathways, we cannot interpret this. But fluorobenzoate degradation, this is activated in the FMT, before FMT sample. So the fluorobenzoate degradation is known to be activated in the Crohn's disease patient. So we can guess that this pathway is a cause of severe condition of the intestine of the patient. So we can find that bacteria. They have the genes related to fluorobenzoate degradation. So this Venn diagram shows the species of bacteria. They have the genes related to fluorobenzoate degradation. So it is immediately find that only before FMT sample, those Klebsiella or Escherichia or Salmonella, they have the genes related to fluorobenzoate degradation pathways, but removed by FMTs. So if we can eliminate such kind of bacteria, so we can mimic the mechanism of the FMT, but it is very difficult. It is very difficult because the antibiotic is not specific to each bacteria. So they have the wide range of spectrum. So we focus on the viruses living in our gut, intestine. So we know that not only bacteria lives in our gut, intestine, but also viruses, many kinds of viruses live in our intestine as well. But those viruses cannot infect to our cell. but can infect to bacteria. So such bacteria is called bacteriophages. So the researchers in the field of microbiome know that there are unknown sequences in the metagenome data of bacteria in our gut. But if we search for the observed sequence to a database, so no hit. Yeah, because the databases of bacteria are very rich. However, the databases of the bacteriophages was very poor. Yeah, when we started the researches of the bacteriophages 7 years ago, at that time, so researchers know that in the metagenome data, the virus-oriented sequences are contaminated. However, the researchers cannot put the annotation to those sequences, so they called viral dark matter. But we are very much interested in the bacteriophages, so they can infect to bacteria very specifically. And please remember that there are roughly 2 types of bacteriophages. Yeah, one is called lytic phages, and the other is called lysogenic phases. So the difference is just one point, that the lysogenic phages, so both bacteriophages insert their genome into the bacteria. However, the lysogenic phages, their phage DNA are integrated into the bacterial DNA and transcribed and assemble to phages and break the cell membrane of the bacteria and go to outside. However, the lytic phages only increase their number and assemble and break the membrane of the bacteria and go outside. That's it. So the difference is integrated or not integrated. But the lytic phages had the history of the used as a therapy. It's called phage therapy. So pathogenic bacteria, the bacteriophages is used as a therapy in West Europe countries, including Russia. And also in US, the US San Diego has a special institution called IPATH to investigate the bacteriophages as a therapy. 7 or 8 years ago, we focused on the bacteriophages. So we create an experiment protocol in order to extract the bacteria DNA and also a bacteriophages DNA from the same fecal sample. And we can have the sequence data of the bacteria and also bacteriophages from the same fecal sample. So, but the bacteriophages sequence data, it is very hard to put the annotation at that time. So we create an analysis pipeline in order to classify the observed sequence data of the bacteriophages. So after development of the analysis method, we can have the composition data of the bacteriophages like bacteria. So a single strand DNA phages or double strand DNA phages can be sequenced in the same time. But the bacteriophages researches were performed in the field of not medicine, but agriculture in 1990s or 80s to 90s. So 20 or 30 years ago, at that time, the researches of bacteriophages are very hot topic. However, in the medical field, the very small number of researches were performed. So we don't know the relationship between the bacteriophages and the diseases. However, when we sequence the bacteriophages in our gut microflora, so novel bacteriophages, many number of novel bacteriophages we discovered, and also we observed the bacteriophages data and the bacteria data in the same fecal sample. We can identify the host-parasite relationship. Host is a bacteria. Parasite is a bacteriophages. The infection relationship can be found. So please remember that lysogenic phages, it integrates their genome into the bacterial genome. So in the bacterial data, we can identify the bacteriophages integration site. So host-parasite associations can be found like this. And also lytic phages, they don't integrate their genome into the bacteria. However, by looking at the CRISPR loci of the bacteria, so the history of the infection of the bacteriophages are remained in the CRISPR loci. So I would like to skip the detail. However, finally we create a first database of the 100 healthy Japanese people's bacteriome and virome data, and also host parasite association. So after creating the databases, we search for the bacteriophages that can infect to Clostridium difficile. If we can identify the bacteriophages that can infect to Clostridium difficile, it can be a therapy. It can be a therapy. However, for bacteriophage therapy, the lysogenic phages cannot be used. Only lytic phages can be used for bacteriophage therapy. Because the lysogenic phages can derive the undesired genes. Yeah, for example, the Escherichia coli 0157. This is a very famous pathogenic bacteria, and it has a toxic gene called verotoxin, and also Shigella. This is also a very famous pathogenic bacteria, and the toxic gene is called Shiga toxin. The two toxins are different name, but the sequences of the two toxins are almost the same. So it is considered that the bacteriophages propagated the toxins genes. So by this reason that lysogenic phages cannot be used for phage therapy. So firstly, we search for lytic phages that can infect the Clostridioides difficile by looking at the CRISPR loci of the Clostridioides difficile. However, we cannot find the lytic phages. No lytic phage can infect the Clostridioides difficile. However, the many lysogenic phages, their genome were integrated into Clostridioides difficile genome. So we gave up to use bacteriophage itself. However, those lysogenic phages integrated into Clostridioides genome, they assembled to bacteriophage form and produce some enzyme to break a membrane of Clostridioides difficile peptidoglycan from the inside of difficile. This enzyme is called endolysin. So we investigate the region of the integrated bacteriophages DNAs in the Clostridioides difficile and find 10 novel endolysin sequences. And we produce these novel 10 endolysins by using the E. coli, and we perform the infection experiment using mouse, and by infecting Clostridioides difficile, the more than 70% of mice were died, black curve. However, after infecting the Clostridioides difficile, we treat with our produced endolysin to infected mouse, so they can survive, not died, with a statistical significant difference. So we don't use the bacteriophage itself, but use the enzyme produced by bacteriophages, and that break a membrane of the bacteria, pathogenic bacteria. This is called second generation bacteriophages. We call it. So not only cross-region difficile, but also other drug-resistant bacteria. This is a world threat.
Definitely want to put an H there.
Yeah.
It's not a treat. But
Yeah. Yeah, well, WHO estimates the number of deaths by drug-resistant bacteria will be larger than the deaths of cancer in 2050. Yeah. So we need to create effective countermeasure to such drug-resistant bacteria. Yeah, this is the last slide, just references. Yeah, thank you very much.
Thank you so much, Dr. Imoto. It was very interesting.
thank you. Very, very interesting area. I just have two comments that support, well, one of them supports your work. We have a study, well, myself, my colleagues have a study coming out showing that antibiotic use within 60 days of immunotherapy significantly reduces immunotherapy response due to the reduction in the colonic microbiome diversity. So the colonic microbiome has amazing, spectrum of effects. The other thing that came to mind is there was a recent controversy or pitfall noted that in microbiome studies, which is that Illumina sequencing protocols at some point use the Phi-X 174 phage. And some sequences in the databases are contaminated with Phi-X 174 sequences. So it can cause like a spurious, an artificial match with in the microbial genomic databases. So we, yeah, so.
Yeah, thank you. Thank you. Actually, in the not only a human, but also a bacterial reference genome, there are many bacteriophages, genes, the DNAs are contaminated. So we need to care about this very much. Thank you.
Thank you for such a fascinating study. In the introduction, you mentioned the almost 20 million genes from the bacteria.
Yeah.
But it sounds kind of overlapping. The function-wise, they maybe this is counting from different bacteria, but the same function, same protein, enzymes.
Yes, right.
Counted separate genes. So considering that, how many actually the genes or how many proteins actually over?
Yeah. Yeah, actually, this number is referenced by to some papers. But when I analyzed the 1,000 microbiome data, the metagenome shotgun data, and we identify 12 million genes and non-redundant. The similarity between the ORF is maybe less than 60%. 60%, yeah. But when we search for those ORFs to protein databases, but only 40% are annotated. So remaining that 60% are not annotated. So it means that functional unknown. So our analysis is just the part of the known ORF.
Thank you. In that regard, so it's really hard to figure it out based on the gene
Yeah.
function. Oh.
Any other questions? Can I ask one question? So when you transfer the, you try the FMT, I saw that most of the pairs, the donor's profile took over. I wasn't sure how long it lasted, but some of them did not get replaced by the donor.
Yeah, like this,
What-
for example, the
Right.
third patient after FMT, they're not similar to the donor's pattern.
Or FMT6, like I think, oh,
Six.
I'm sorry,
FMT6,
was taken over.
yeah.
FMT2 wasn't taken over.
Yeah,
What do
taken
you-
over. Yeah, gamma proteobacter taken over. Yes, yes,
What
right.
do you think? Is it more on the donors, something in donors,
Actually,
features or
yeah,
combination?
actually, if we have the more samples, we integrate the metadata of the patient and the donors, and but only that's 9 patient data. Yeah, it is very hard to perform such analysis, and also the period after FMT is not controlled. There's some difference there.
Great. I was just wondering if it's like a host or the, like
Yeah.
the bacteria or donor or recipient. So it's very interesting. Thank you. Any other questions?
But thank you very much. But I really felt that gene technology, you can analyze tremendous information with the modern technology. So it's like in the old days, you isolate bacteria. If you can't find it, and the report comes back, we couldn't detect it. But nowadays, you just bypass it. You make a whole vegetable juice or something, and you analyze the content, and you don't care if it's carrot, if it's tomato, but you just analyze the content of the genome, and you can talk a lot, and you can intervene. You can make a tremendous amount of intervention in every field: bacteria, testing, and also cancer research, and everything can be done in the level of gene, even bypassing all the intermediate structure. So it's a huge paradigm shift in the technology by using the technology.
And additionally, the composition patterns can be affected by a SNP pattern.
So all the presentation are very affected or related to each other, and it was very, very wonderful. And I hope in the future maybe you can give us a kind of capsule to take it to make everybody healthy. Thank you very much for a nice talk.
Thank you.
So now, thank you. Give him a big.